Hello, everyone. It's a great pleasure to introduce you Pascale Vonaesch, who's visiting us for 2 days here at IGC. Also, thank you, everyone online. Pascale, she's a young group leader. I think she has her lab for almost 3 years now, and she's at the University of Lausanne. She did her PhD in ETH Zurich with Dietrich Starhart, studying Salmonella host interactions. And then she went to a postdoc at the Pasteur Institute with Philippe Sansonetti. And as I just mentioned, she's now has her lab at the University of Lausanne, where she's studying the effects of malnutrition and interaction with the microbiota, but I'll let her tell you about it.
So thanks a lot. Thanks a lot to all of coming, and thanks a lot also for Karina for the invitation and give me the opportunity to share with you today a few of the recent studies that we did on the microbiome and childhood undernutrition. So, oops, doesn't seem to work anymore. Maybe. Oh, now it works. Perfect. So I would like to start with the graph that is showing basically the evolution of the infectious diseases over the last 50 years. And as you can see here, there has really been like a very steady drop in many of the infectious diseases from the 1950s on, mainly induced by the use of antibiotics and also by vaccines. However, and quite importantly at the same time, we also had an increase in many non-communicable diseases. So Crohn's disease, also multiple sclerosis, that really on a global scale increase in their prevalence. And what's quite interesting is that many of these non-communicable diseases are actually linked to the microbiome. So many of you are probably familiar with the microbiome, which is the community of microorganisms that live in and on our body. We mostly talk about bacteria, but the microbiome also comprises actually microeukaryotes, also viruses and fungi. And in our body, the densest composition is found in the intestinal tract. And here you have a little sketch where you see that this microbial community is separated from the underlying tissue by different barriers, which include a mucus layer and also different measures of the immune system. And as you all know, the microbiota is also really important in our health, especially in the microbiota host crosstalk and in immune maturation in early life. The microbiota is also implicated in the production of vitamins and in proper digestion, and it's therefore really important that we have a healthy microbiome to be in good health. So the intestinal microbiota is obviously important for functions in the intestine, but it's also important for axes that basically link with the intestinal microbiota. So one axis that you maybe heard about is the microbiota gut-brain axis, where we have especially metabolites from the microbiota that have effects on brain health. There's another axis which is basically the gut microbiota liver axis, which is important especially in metabolism and also like in undernutrition. And there's another axis that isn't indicated here but was a lot in everyone's thoughts lately, which is the gut microbiota lung axis, which was also important as an example in the recent COVID pandemic. So if we now have actually what we call a dysbiosis, the whole situation is a bit changing. So the microbiota composition is already changing. We have more aggressive bacteria which take over. They're able to breach these barriers and get access to the underlying tissue. And with that, we have then tissue destruction and at the same time also chronic inflammation. And such dysbiotic diseases are also associated with many nutritional diseases, and notably with one of the nutritional disorders that my lab is most interested in, which is stunted child growth. And stunting, as we normally call it, is a reflection of underlying chronic undernutrition and is indicated actually when a child is more than 2 standard deviations below its normal growth. So stunting is very widespread worldwide. It's about 1 out of 4 children under the age of 5 that is affected. And it's especially prevalent in sub-Saharan Africa and also Southeast Asia, as you can see in this graph, where you have actually like the most affected places indicated in dark red. And it's not so important actually the size per se. So you would say, well, someone is small, so what? And indeed, so what? But what's important is the associated consequences, which include a higher morbidity and also diminished cognitive development. And quite importantly, also the fact that the children that are undernourished early in life, they have a higher risk of obesity and also of cardiovascular diseases in later life. So as you might know, there's several risk factors for undernutrition. One is obviously poverty in all its different aspects. Also inappropriate weaning food, an unbalanced diet, and macro and micronutrient deficiencies, and also repetitive infections. And you see here a few pictures that I took in Africa where you can see that basically like there's quite a specific diet that the children often receive, especially in poor families with a lot of starch. And there's also a lot of infections, so you see one of the study areas that we worked in where you see that there's really a lot of problem with sanitation. However, even knowing that, and even by giving basically these children very sophisticated treatments, which include micronutrients and also like a very balanced diet, it isn't really possible to correct for the encountered growth delay. And there was the question actually, why is this really the case? And recent evidence then also said, well, it is something that is happening around weaning, because if we look actually when there's really this decrease or this worsening in stunting, it's happening right after the weaning period. But the question still remains, so it's happening after weaning. We don't manage to basically do it with nutritional interventions alone, so what is going on? And one idea was that the microbiota and also the gut ecosystem could play a role and basically be an additional element in this equation that hasn't been taken into account so far. So first evidence came now almost 10 years ago from a group in the United States headed by Jeffrey Gordon, and what they did is that they basically used fecal samples from children that were either undernourished or not, and they transplanted them in germ-free mice, so in mice that don't have a microbiota on their own, and they then colonized one group of mice with feces from healthy children and one group of mice with feces from undernourished children. They then exposed both of these groups of mice to what they call a Malawi diet, which is a diet that is really depleted in micro and in macronutrients. And what they observed is that the mice with the normal microbiota remained more or less normal, but mice that had actually like undernourished microbiota lost a lot of weight. So they then went on and re-nourished both of these groups with the food that these children normally receive when they are in therapeutic programs. And what they observed is that both of these mice were actually like, again, getting weight, so it's the same thing happened as with children. You re-nourish them and they basically get better. But the big surprise came when they put them back on the Malawi diet, because while the normal mice still remained normal, the mice that had the undernourished microbiota again lost weight, showing that there was actually like a causal and direct role of this microbiota mediating that they basically weren't really able to be re-nourished with this food alone. So this was really interesting and was then followed by a second publication of the same group where they went on and also looked what happened now if you co-housed the mice that were undernourished with the mice that were not undernourished, so basically the ones with the undernourished microbiota with the other ones. They put them together in a cage, and what mice do is that they basically eat each other's feces, and they then have what you, like a natural exchange of microbes. And what they observed is by doing so, the mice that were formerly undernourished, so had the microbiota from the undernourished child, were suddenly doing much better and were actually gaining better growth. And when they looked a bit more in detail in the microbiota, they could see that basically there were some bacteria from the normal mouse that were invading the undernourished mouse and therefore basically conferred them a growth benefit. So what they showed was that there indeed are missing taxa in these undernourished children. And basically, like this missing taxa are having an important function. What remained really puzzling is actually like that it wasn't really clear what the molecular mechanisms were, so basically what this bacteria were doing. And this is where our own work with the Afribiota consortium came in. So we were wondering actually what changes we could find in the gut ecosystem of these stunted children. We were also interested to know if basically these are changes that are the same across different study sites or if they're basically specific to a given context. And we were also interested to know if they're causally implicated in the pathophysiology, which would then help us to actually design better treatments to really modulate this microbiota for better health. So for this, we set up a consortium together with Institut Pasteur, where I did my postdoc and initiated this project, and also together with Institut Pasteur de Bangui and de Madagascar, which are two Institut Pasteur in two of the countries that are most affected by stunting. And what we then did together with the whole field team is that we recruited children in 3 groups, so normally nourished children, also severely and moderately undernourished children. And we screened actually like almost 2,000 children, and that included 920 children And for all of these children, we took different samples. So we had blood, urine, and also feces to look for biomarkers and immune cells. And also we took gastric, duodenal, and fecal samples to profile the microbiota across the gastrointestinal tract. And here I just put you a few pictures to show you how such a study is really looking like. So a lot of work was actually just going and explaining also the community what we were doing and what the microbiota is and why we needed basically the samples. And another big thing was also to take this duodenal sample. So this was the first time this was done. And you need actually to introduce a small tube into the duodenum, which was something that Professor De Marquez that is here was really training the doctors on to make sure that we didn't have any problems by doing so. So we then did more or less classical microbiota analysis, both actually by culture for the duodenal samples and also by sequencing. And in the first time, we did amplicon sequencing with 3 different amplicons, 16S for bacterial and archaeal taxa, 18S for eukaryotes, and ITS2 for the fungal composition. As said, we also had something that was quite special, that we looked actually at the upper and the lower gastrointestinal tract. And why is it important actually to also look at the upper gastrointestinal tract is that we know that basically the ecosystems aren't really the same. So basically like the living conditions across the different signs are not the same. With that probably also the bacterial communities, and especially nutrient absorption takes place in the small intestine. So if you look in the feces, you're basically looking at the microbiota composition that is a bit well, which is kind of at the wrong place. So as said, the composition is a bit different, and it's not just the composition that is different, but also the whole profile. So you see as an example that there's a difference in which nutrients are available along the gastrointestinal tract. You also have changes in the pH, which is changing actually from the small intestine where you have influx of bile, and it's basically then different in the feces. Oxygen concentrations are also higher in the small intestine, and quite importantly, you also have a different bacterial load with the small intestine, which is quite loosely populated, and then basically the feces, which are having much more bacteria per gram of feces. So what did we see when we did this analysis? We first started by looking at the duodenal samples, and here the first question that we had was really to see if we had more bacteria in the small intestine that we should have. This is a phenomenon that is called small intestinal bacterial overgrowth, and is associated with many different health problems, including a so-called leaky gut, also a decreased peristalsis, and a pro-inflammatory state, and often it's also associated with a malabsorption. And what we observed in our children was that indeed we had actually many of these children too many bacteria in the small intestine, so many of the stunted children were suffering of the small intestinal bacterial overgrowth. And here you see actually the table with the actual numbers of bacteria that we detected. Here you have the detection limit of what is considered normal. And you can see that here, especially in Madagascar, in many of these children we had 10, 100, or for some of them even 1,000 times more bacteria than what they should have. So they really had a real purulation of bacteria within the small intestine. So the next question was then obviously to say, who are these bacteria? So who is leading to this bacterial overgrowth? And here we had a really big surprise, because we expected to see like the classical SIBO, so basically like fecal bacteria, but instead we found that we had actually like about 60% of all of the bacteria that were bacteria that are normally found in the oropharyngeal tract, so basically like oral bacteria. So this was really astonishing. We then also looked in the feces of these children, because for the duodenal samples, for ethical reasons, we could only take samples from the stunted children, while we had feces actually for the stunted and the non-stunted children. And when we compared the profile of these two groups of children, we could see that in the stunted children, we didn't only have SIBO, but we also had higher levels of this oral bacteria in the feces in the stunted compared to the non-stunted children. So this really indicated that basically this SIBO and this oral overgrowth seems to be something that is specific to the stunted children. From there, we were then also interested to know what the consequences of the SIBO really were. And as I initially told you, one idea was that it could actually lead to inflammation. And we therefore measured pro-inflammatory cytokines right in the duodenal samples of these children. And you see here actually like a profile of significantly differently present cytokines, with IL-6 and MCP1 and other pro-inflammatory cytokines being really more present in the stunted children, indicating that these children actually were suffering of small intestinal inflammation. We then also compared the profile of bacteria and looked for this oral bacteria and the presence of other bacterial groups, as an example here indicated in blue, intestinal bacteria. And what we observed was also quite interesting. So what we saw is that many of these oral bacteria co-occur together. So basically like if you had one type of oral bacteria that was basically there, it was quite likely that you also had other oral bacteria present. But on the other hand, they were co-exclusive with intestinal bacteria, meaning that if you had oral bacteria that were overgrowing, they were kind of pushing away basically the intestinal bacteria that should normally reside there. And this is something that hasn't been described before in undernutrition, so this was really exciting because it basically showed us that there seems to be really something specific, but this displacement of oral bacteria in the small intestine is something that was found already in IBD, in intestinal bowel disease, which is also an inflammatory condition, so it was something that really seems to be a recurrent theme. So we then also looked in more detail who these bacteria really were and looked at the core small intestinal microbiome. So here you see actually first a frequency plot where you have the main phyla indicated with different colors. And you can see that except actually for the proteobacteria here indicated in red, most of the phyla were more or less the same in between the study countries. And when we looked for the core microbiome in between the two different countries, Madagascar here in green and Central African Republic here in blue, we could see that actually the vast majority of the core microbiota was really the same in between the two study countries. And what was even more exciting is that there was also a study coming out 2 years later by, again, the group of Jeffrey Gordon. And what they observed is that they had exactly the same list of bacteria that they found in stunted children in Bangladesh, meaning that basically different study size and different studies per se were really always falling back on the same bacteria that seem to be overgrowing in the intestine of these children. So in this paper that came then out by Jeffrey Gordon, they also started to look at the pathophysiology. And what they saw is that they saw actually that there was a correlation in between the stunting, so how much actually the child was too small, and the total bacterial load in the small intestine. And they could also see that they had a direct correlation between some of these oral bacteria, as an example, Veillonella, Streptococcus, or also Rhotia, with the absolute level actually like of stunting, meaning that these oral bacteria seem to be directly correlated to the degree of stunting. They then also performed some mouse experiments where they basically took isolates that they had from the children and put them again in germ-free mice. And even though I don't think this is the most convincing part of the paper, they could show that basically this bacteria are leading to some kind of inflammation when they compared it to mice that were colonized by bacteria that they isolated from mice. So this was the first indication that these bacteria really have a direct role in the pathophysiology. And we were then wondering if basically like this bacteria maybe had other roles. So inflammation for sure also can lead actually to undernutrition, but we wondered if maybe this bacteria would also be implicated in nutrient absorption. And to address this question, we then used two different approaches. First, we actually took the bacteria that we isolated from the stunted children and co-cultured them with small intestinal cells. And at the same time, we also took the bacteria and gave them actually to mice to then directly assess in the mice if we could see any changes in nutrient absorption. So here just to show, for the cell experiments, we used two controls. So we had a positive control, which was Lactobacillus paracasei, that is known actually not to influence lipid absorption. And at the same time, we also had the negative control, which was a marine E. coli, which is known to lead to decreased lipid absorption in epithelial cells. So what we then found was that indeed, as expected, the positive control did not affect lipid absorption. So here you have lipid absorption in the cells normalized to control cells. And as expected, we also had a strong decrease in lipid absorption induced by Escherichia coli. very nicely, when we then looked at the oral isolates that we had from these children from the duodenum, we could see that all of these different bacteria that lead to this overgrowth in the small intestine were actually at differing degrees leading to a decreased lipid absorption, showing that there seems to be a second mechanism what this bacteria are really doing in these children. We then went on and also characterized Streptococcus salivarius isolate that had the biggest effect on lipid absorption a bit more in detail. And what we saw is that basically, again, we saw a decrease in lipid absorption with the whole bacteria, but we also saw a decreased lipid absorption if we took either whole supernatant or filtered supernatant, indicating that the small metabolite was probably mediating this effect. And as a control, when we then took actually bacteria that we heat inactivated so that are not metabolically active anymore, we didn't see any decrease in lipid absorption, and we also didn't see any changes. We just acidified the medium to mimic actually the production of this fermentation product that is happening in the context of Streptococcus salivarius. So this indicated us that there seems to be a small metabolite that is really the culprit for this decreased lipid absorption. But the question was, who could this now be? And just a year before, also in the group of Philippe Sansonetti, Joao Araujo found out that there's basically lactobacillus and also E. coli can both influence lipid metabolism in epithelial cells. And what he found is that actually E. coli is doing so through acetate, while lactobacillus is doing so through lactate. And he could show that both actually then had less lipids in the systemic site, but that basically only E. coli was really decreasing the lipid absorption within the cells. So what we then did is that we compared the metabolites produced by our Streptococcus salivarius with lactobacillus, and first looked at acetate and lactate, but basically there was no changes. So there was as much lactate produced by lactobacillus and Streptococcus salivarius, and acetate wasn't really produced by Streptococcus, so that didn't seem to be the same mechanism. From there, we then went on and basically measured 74 different metabolites to see if we could find a metabolite that would be differentially produced by the different strains. And indeed, we found 15 metabolites that are differentially expressed. Here you have a few examples with 2-octoglutarate or 2-octoisovaleric acid that are basically produced by Streptococcus but not by lactobacillus. And right now we're measuring all of these metabolites to find if we basically can really boil it down to a single metabolite. And this is work that is performed by my postdoc Carla, and hopefully in a few months we can then tell you what we found and who is really the responsible. So from there, the second question was obviously also not just the mechanism within the cell and the mechanism from the bacterium, but also to know if this also holds true in vivo. And for this, we used actually a mouse model where we pre-treated mice with antibiotics and then basically gave Streptococcus salivarius or lactobacillus as the control strains to these mice. And we then basically gave to this mice a bolus of sunflower oil that we mixed with the fluorescent dye, and with that we could then actually afterwards see how much of these lipids were really absorbed within the small intestine of this mice. And here is just to show you that basically our treatment here really depleted almost all of the microbiota, so these mice are quasi monocolonized actually either by Streptococcus or by lactobacillus. So what we observed was indeed very nice, because here you have again the control mice that didn't receive any bacterium, then Streptococcus and lactobacillus, and as you can see there was a very clear decrease in the lipid absorption in the presence of Streptococcus. And this was also the case actually systemically, so basically when the mice were gavaged with Streptococcus, they also had less of the lipids that reached the liver, meaning that there was really a clear effect of this oral bacteria in lipid absorption, both in the intestine and systemically. And what it also told us is obviously if we renourish these children or these mice, but at the same time they're not able to absorb the nutrients, obviously this will not really work, and so this overgrowth of oral bacteria really seems to be part of the problem. So now I told you a lot about the upper gastrointestinal tract, but we were still interested to know also what is happening in the lower gastrointestinal tract. And here, actually, what is known already since a long time, and where just a very nice review came out, is that in the context of dysbiosis, we have different changes. So we have, as an example, higher levels of oxygens, we have also higher levels of nitrate, and we have changes in given metabolites, including actually in the profile of bile acids and also in short-chain fatty acids. And all of that is then basically leading to a leaky gut, so impaired gut barrier, which makes the bacteria can translocate and also leads to systemic inflammation. So we were then interested to know what we could find actually in our children. And here you see basically the results of the comparison of the feces of stunted and non-stunted children in terms of bacteria. And you can see that we have a whole group of bacteria that is less prevalent in the feces of stunted children compared to the non-stunted controls. And if we look a bit more closely who these bacteria are, many of them are actually members of the class of Clostri-TOF, which is a group of bacteria that is known to basically degrade poly and oligosaccharides into the short-chain fatty acid butyrate, and also to convert primary bile acids into secondary bile acids. And we went then on and also assessed for bile acids in the feces of these children, and quite nicely we could see that indeed there was a decrease in the secondary bile acids in the context of stunting. We then also wondered if this was really correlated to the microbiota, and by correlating specific groups of microbes with the levels of this secondary bile acid, we could see that there was a nice correlation, as an example, by different members of the Lachnospiraceae and the levels of secondary bile acids. So there seems to be really like changes in the gut microbiota also of the stunted children, and the next question that we wanted to ask was then also to know if there were not just like general microbiota changes, but also changes in enteropathogens, because like since a long time it's suspected that infections play a big role. And indeed, there were several publications coming out last year where they have shown that basically like the number of infections, either bacterial and viral or parasitic, was correlated with overall gut inflammation, both if it was basically like a symptomatic infection, but also if it was a non-symptomatic infection. This was also shown in another publication from another team. And we could confirm the same thing also in Afribiota, showing that basically like the number of virulence genes that you would detect in the fecal samples, so how many different pathogens you have, this would actually directly impact the inflammation of the gut. However, in our data and also in other data, it was not really clear if there was just an association between pathogens and inflammations, or if there was basically also an association with stunting. So it seems that there's basically different things. Stunting is not necessarily associated with inflammation, and inflammation is also not necessarily associated with stunting, even though this was initially was thought. So the last point we then looked at were the microeukaryotes. So I told you in the beginning that the microbiota is composed of different microorganisms, the bulk of the biomass being the bacteria, but also a smaller subset coming from microeukaryotes, including fungi and different protists and also helminths. And therefore, we wondered if basically like there could also be changes in these microeukaryotes that could act as keystone species and therefore have a direct effect on stunting. So we first started and looked at the fungi in the different compartments of the gastrointestinal tract. And quite interestingly, we could see that as for the bacteria, there was a clear difference actually between the upper and the lower gastrointestinal tract in the composition of this bacteria. As was also the case for microeukaryotes, where we had quite a large variation actually in between the different subjects. We could then also see that we had a signature that was really specific per country. So basically looking at the microeukaryotes, we were really able to distinguish the two study countries with a specific profile being found in Madagascar here in red, and a specific profile being found in the Central African Republic. However, came the big disappointment when we then looked actually at the association with stunting, because when we looked at first alpha diversity, so just how many different parasites we had, there was strikingly no effect on stunting status, not with the fungi and not with the microeukaryotes, no matter how we analyzed the data. The same was also the case when we look at beta diversity, basically, so how much the samples were different in between each other. And the same was also true when we looked at prevalence. So basically, there was not really an association between the presence of given microeukaryotes or the presence of given fungi. And this really told us that it seems that the bacteria are really the ones that are driving malnutrition, and at least in our study context, the microeukaryotes don't seem to play a big role. So in conclusion, for the stunted child growth, it seems that there's really two different entities that are contributing to this phenomenon. First, there's a recurrent exposition to pathogens, and there's either specific pathogens or the general enteropathogen burden that is directly contributing to malnutrition. And at the same time, we also then have changes in the resident microbiota, so dysbiosis, both in the upper and the lower gastrointestinal tract, that seem to be directly and also causally implicated in the syndrome. So there's a lot of outstanding questions still there. So first, we still don't really understand how this mechanism is happening. We could boil it down a bit more to say that it's probably metabolites produced by these bacteria that are implicated in the pathophysiology, but we still don't know how this is working on the host side or also on the bacterial side. Then we also don't really understand why we observe SIBO to start with. So there seems to be this big overgrowth, but it doesn't seem to be dependent on stomach pH. So one of the big questions is, what is it then? And for that, we're working right now together with Aaron Elinav to also look at AMP profiles and see if maybe a change in antimicrobial peptides would be the culprit that allows for overgrowth of this bacteria. And last, there's also not clear yet if basically like these changes are having consequences on the immune system, and also if they're basically like changes which are specific to given places, or if there's changes that are basically like more global. And the last and most important is also the question, so we know now that this oral bacteria seem to play a role in this disease, but how can we now really intervene? So with that, I would like just to end with a few slides on what we're doing right now, which is basically trying to develop microbiota targeted interventions to really correct for these diseases. So I told you in the beginning that this dysbiosis basically is associated with changes in the microbiome that then lead to all of these pathophysiological changes. And obviously the big challenge is then to bring this back and bring it back to a homeostatic state where we have a normal microbiota composition and are also able to renormalize the pathophysiological consequences. So in the past and right now for different dysbiotic diseases, this can be done by different means, either by antimicrobial substances, by probiotics, which are live bacteria, symbiotics, which are a combination of fibers and probiotics, also postbiotics, which are bacterial metabolites, or fecal transfer or phage therapies. And, oops, there was a problem here. And right now we are actually like working together with Lisa Meyer at the University of Tübingen and with her postdoc, Nermin, on finding drugs that would really be able to basically like suppress the bacteria that we don't want to be there, while basically not touching the bacteria that we believe are good. And in this project that is funded by the Bill and Melinda Gates Foundation, Nermin is first screening basically against reference strains to really like nail down which are the important drugs that could basically have a role. And she will join us early next year in Lausanne to also like complement this data set to see if these drugs are also working on the actual isolates that we have from these African children. And obviously the end will then be to basically test these drugs also directly in communities to see if these drugs will really be able to shift not only like individual strain, but also like whole communities. So another aspect that we're pursuing in the lab is also to look at next generation probiotics. So next generation probiotics are, in comparison to the normal probiotics that are normally isolated from fermented foods, substances that are isolated from healthy human subjects. And the advantages that we think that they have is because they're basically more physiologically relevant, they should have an easier time to really establish them in the gut. So if you basically think about it, for the normal probiotics, it's a bit like you would take a palm tree and basically put it in a forest in North Europe, while here you would basically like take a plant or something that is already a bit adapted to the environment and therefore has it much easier to get implemented. So these next generation probiotics are basically quite like a hype at the moment, but they're also like associated with a few issues. Because what you have to do is basically first to isolate them from healthy subjects, characterize them, and then grow them up. But this identification, even though basically like it sounds really easy, is not that easy because you have to identify like thousands of clones to really find the one that you're interested in. So how this is done right now is that basically a lot of people are just using sequencing, which is time consuming because you have to isolate the DNA from every single strain and then sequence it. It's also quite cost intensive. And at least for like strains that are coming from the clinics, there's also an easier way where you can use a MALDI-TOF system where you basically pick a clone and can then identify the bacterium based on a metabolic fingerprint in a few minutes. However, there's a few issues. So basically, like with this MALDI setup, even though it's relatively cheap and performed within minutes, we're still missing a lot of databases, and because it has been developed for the clinics, there's especially databases that are missing for commensal strains. So this is something that we tackled with my lab while still in Basel, and also with the help of different clinical microbiologists, which provided us with clinical isolates. And with that, we put together a bacterial culture collection of 142 strains, which covered 47 species and 21 genera, which are the main representatives of the commensals living in the gastrointestinal tract. We then acquired spectra for all of these strains and built a MALDI library that could then be combined with a commercial library and be used to basically identify these isolates. We now characterize also this library in quite some detail and applied it also to a strain bank that we have from Switzerland. And as you can see here, it indeed seems to work. So if you basically take these isolates and you just like use the traditional MALDI library, we're able to identify roughly half of them. But if you use now our new library, we're able to basically like significantly increase the number of strains that we are able to identify and basically just have like a few ones that we're not able to identify anymore. So I think this is really promising because it tells us that we can really use also like these tools to speed up the identification of this bacteria and basically then use them for medical use. So with that, I would like to close just to say you why this is actually important. I made quite a jump from basically telling you that there's changes in the microbiome to telling you about the tool that you say, well, but what do we do now with it? And just a few months ago, there was a publication coming again from the Jeffrey Gordon lab where they showed for the very first time the use of a probiotic, a next generation probiotic in undernutrition. What they did is that they saw that there was a decrease in bifidobacteria in stunted children. So you see here in red the evolution actually of the relative abundance of Bifidobacterium infantis in stunted children and in blue in non-stunted children. And what they did is that they then ectopically gave these children not only renutrition, but they mixed it with a symbiotic strain of Bifidobacterium infantis that was isolated from a child in the United States. And indeed, when they then looked at the weight increase in these children, what they could see is that they had a small but significant increase in weight and in height, meaning that this Bifidobacterium was really able to boost the growth of these children. And this was really promising because it showed that the microbiome seems to be one of the missing parts of this equation, how to better treat these children. And I think what was also really interesting is that they also basically showed that this bacterium was colonizing, but that basically in a mouse model, a strain that was coming from Bangladesh was even able to colonize better. And I think this really raised the question about personalized interventions. So basically, is it really possible to basically just isolate a strain in Switzerland or in the US and then try and transplant it to an African child? Or do we actually really need to locally adapt the interventions that we have? And I think based to that is also the other questions, are the signatures that we see really global, so as we saw for the small intestine, or are they basically also locally adapted? So basically, do we first have to study the microbiome in every context and then basically adapt the treatments to the specific context? And here I don't want to go in a lot of details, but this is just to mention that we now have a study in Ethiopia, in the Somali region, where people have a completely different staple food, drinking especially a lot of camel milk. And these communities have a microbiota that is completely different from all the different microbiota compositions that we have seen so far, with especially also a lot of streptococci and also bifidobacteria. And we're right now characterizing also the microbiota in a bigger cohort to basically see if we would see the same signatures in stunting, or if the stunting signatures are also geographically specific. And with that, I'm at the end of my presentation, and I hope I could show you that non-communicable diseases are really on a global rise, and that treatments so far are not very effective. I could also show you that this is probably the case because we still miss the microbiome in the overall equation of these non-communicable diseases, and that especially in stunted children, there seems to be specific changes to the microbiota that are associated with the syndrome. Then I also showed you that these changes are causally linked with the pathophysiology, and that therefore basically to better treat childhood undernutrition, we should really aim not only to restore nutrient intake, but also the overall microbiota composition. And as I mentioned, there are several microbiota targeted interventions that are currently developed to improve the nutritional rehabilitation scheme. And I think there it's actually like important to say there's still a lot of questions that remain. We made a big jump, I think, in the last 10 years to understand what's really happening in the gut of these children. But there's now as many questions that re-emerge to know if this is basically globally applicable, and also how we can really modulate the microbiota to have a better health for these children. And with that, I would like to thank the whole consortium that was involved in Afribiota, also my group in Lausanne, our founders for Afribiota and for our current work, and obviously also you for your attention. Thank you very much.
Thank you, Pascale. And so just before the questions, I also wanted to say that at 4 o'clock, Pascale is will be talking with postdocs and students about her career and advice if you want to join in the next room. And there's also slots for lunch tomorrow if more people want to join. So questions?
I have a couple. Okay, so let's go first to the SIBO problem, right? In this show. So is it, if I understood correctly, the bacteria that are causing SIBO in the kids are very different from the bacteria of adults that have these overgrowth of bacteria in the small bowel, right?
I'm not sure if it's a question of children or adults, but what is, well, traditionally SIBO is basically found in some diseases of malabsorption, and there if they characterize a SIBO, they found like intestinal bacteria that basically like grew up, so they, while in the stunted children we see basically like a bacteria that seem to be from the oropharyngeal space.
Right.
Now
so
I'm not sure someone looked in undernourished adults as an example.
Okay, because I have the sensation that, for example, Escherichia coli is very common to overgrow in SIBO conditions in adults. And the list of bacteria that you were showing for the kids, it wasn't there. So
which bacteria?
So the E. coli. I was saying that E. coli is very common in adults that have SIBO.
Exactly. So it's one of the.
So there should be a difference
Exactly.
where in the bag where they are coming from, right? It looks like in the children it's from the mouse, and then the adult might be from the the large intestine, right?
Yeah.
So the other thing has to do with this last part of the bifidus infantis, right? So it is, I think it is thought that these bifidus bacteria, right, when we are born, they are very good degrading the mother's milk, right, in breastfeeding, right? So I was wondering to what extent are the different strains of Bifidus infantis capable of, taking more, grow better in these milk oligo things that they normally consume, if that process has any relationship to the malnutrition correction phenotype. Right?
So what they basically speculate in the paper from the Gordon lab is that they say that basically like the Bifidobacterium from the US is really adapted to human milk oligosaccharides, while the one basically from Bangladesh is also able to degrade other components because they introduce weaning food much earlier.
Okay.
So basically that's maybe also why it's basically colonizing better in this context.
Oh, I see.
Then basically there was just a paper coming out last week where they could see that there's different bifidobacteria, and apparently around weaning there's also like different bifidobacteria that take over that are able at the same time to degrade human milk oligosaccharides, but already also other nutrients that will come in with the weaning food. So I think it's a...
And if can I have one more? So now it's a question about the adaptive immune system and immunoglobulin diversity in the undernutrition. Is there anything known about that?
So we had a study together with a PhD student from the Finley lab that was working with us, and she looked at IgA targeted bacteria. And basically it was quite disappointing because we couldn't really make sense. So there were a few bacteria that were targeted, there were a few other bacteria that were not targeted, but we couldn't really make sense like which ones were targeted and which ones weren't.
Well, Mark, I'll ask a question. Anyone else? So in the case where you have overgrowth of oral bacteria in the small intestine, so in these children, do you think it starts? So you had a bit of speculation of why, but I was wondering if it's related with the problems with the diet that then lead to a lower colonization of the small intestine, and so basically it would be more like under normal conditions, these bacteria in the small intestines are really inhibiting the oral ones to invade, even though in terms of numbers they are not very high numbers, but they could be inhibitory to these oral bacteria.
Yeah, I think that could be one explanation, because basically there seems to be a trade-off. Either you have a lot of intestinal bacteria or a lot of the oral bacteria. So I think either there's kind of a colonization resistance which is gone, so this could be one idea. And the other one is also that basically like maybe the streptococci, they overgrow because they have more simple sugars. So in undernutrition there's also a lot of starch-rich food. So another idea could be that basically the diet is a little bit different and is in favoring basically the overgrowth. And the third one is this hypothesis that it's basically something from the host.
Of the host.
Yeah. But for the moment we're really completely in the dark. Yeah.
Okay. Can you? How do I know? How do I see?
Here they are. So there's someone that says my master thesis is also about microeukaryotes.
So the question
samples.
is,
Yeah.
have you noticed any difference in the composition of samples from urban and rural communities?
Yeah. So we basically just look at urban communities. So it was 2 neighborhoods in Antananarivo in the capital city. And basically like we didn't really notice differences between the different like districts of Antananarivo, but there's huge differences in between each individual. So basically like the inter-individual differences are even bigger than for the bacteria, which may also be one of the reasons why we can't find any associations with clinical factors, because it's just too much variation to start with.
Okay, so I think we're good, and yeah, thank you very much, Pascale, and thank you everyone.